Okay, so I'll just better tell them. Those of you who are staying home, do the problems anyway. Okay, so what I'm going to do today, I'm just going to go through a couple of problems. And I will lead you through the first one. And then you're going to have to do the others. In groups is fine. Okay, so here's a typical triple point problem. So it says I hand you an F1 organism. So I've already done the setting up the F1s. And I tell you it's heterozygous at A, B, and C. That's all I tell you. And then I also give you one that's homozygous recessive. So you can do a test cross. And this one is not X-linked or anything fancy like that. It is just an ordinary, boring, triple point autosomal cross that you're doing. So there's the results you get from this cross. So there's 1,000 individuals that result. And then I got a bunch of questions I ask you, which I'm sure you're all going to have no problem answering, right? So the first question, what are the parental phenotypes? So here's the phenotypes that you got from your cross. And now I want you to tell me which one of those are the parentals. Go ahead.
The 464 and 465.
Yeah, the most numerous ones. It's that easy. You just look for the ones that are most common. Those are the non-crossover types. So it's pretty easy to spot. All right, and the next question was a little trickier. So I ask, what is the arrangement of alleles? And you all recall I told you that was a sort of a term of art. And it just means what is the arrangement of the different alleles on the two chromosomes in the F1 parent? You can imagine that, well, it's ABC on one chromosome and little a, little b, little c on the other one, or it could be some other arrangement. How do you figure that one out?
Identify the middle one first.
You find the which one?
The middle one first.
That's what you need to know. Is sure you need to know that, but you can also get it with just looking at these groups here. One way to do this is you could just say, okay, there's the most numerous one. There's two arrangements there. There's big A, little b, little c, or little a, big B, big C. And that tells you the arrangement of the alleles. That's it right there. So it's like that. In order to tell us the arrangement of the alleles, you don't need to know which one is in the middle yet. So any order will do. You just got to figure out what's on each of the two chromosomes. Okay, now the next question is which gene is in the middle? How do you figure that one out? Remember, I'm going to make you do all the others all by yourself, so you're going to have to volunteer something here sometime.
It's the one that's different from the other.
Okay, the one that's different. What are you comparing? What do you compare to see which one's different?
Like the two complementary strands.
You're going to have to be more specific than that. So we know, for instance, this one is non-crossover, right? That's the parental type right there. What are you going to compare it to? You have to compare it to something out there.
To the least numerous one?
Yeah, you want to go find the most uncommon one you got. which is this guy right here, big A, big B, little c. And you got to ask, okay, which one, if you're comparing those two, which one is the most different? Now, I picked one that's a little less obvious. So here we got big A, and then this one we got little a. Those are different. But we've also got little c and big C here. Those are different. Big B and big B are the same. So B has got to be the odd one. Okay, so which gene is in the middle? B. Yeah, it's that easy. Okay, so like it says there, you compare the rarest to one of the most common ones, either this one or this one, and you figure out which one's a little different, and that's the one that's got to be in the middle. So B is it. Okay, next. Oh, look at this messy question. Right, and like I'm going to tell you, this is good practice when you're solving these kinds of problems. Is reorganize the data, right? So it'd be a good idea to look at this, and for instance, this is particularly pressing if B wasn't the middle one, because here it lists all these show B in the middle. What if C were the one in the middle? Then you would get hopelessly confused. So one thing you want to do is get those in the right order, because it just makes it easier for your brain to figure out what's what. But then the other thing is, we want to organize these into those 4 categories. The non-crossovers, the single crossover between A and B, the single crossover between B and C, and the double crossovers. So one of the things we want to do here is just sit down, get a piece of paper, whatever, however works best for you, and work out who's who in these categories. So for instance, we already figured out who the non-crossovers are, right? This one, and this one. Well, we figured out what the double crossovers are. What are the double crossovers? There's something funny about this data set. Yeah.
Is it 10 and 20?
Then this one's only got one. This one's rare. But there's something funny about this data set, because notice that 1, 2, 3, 4, 5, 6, 7 instead of 8 different types. What that tells you is that there is a missing entry in this collection, and it's missing because it has 0 representatives. So what's the missing phenotype? That should be right here. But we're going to have to give a number of 0 to.
Well, A, little B, big C.
Yeah, it's got to be the complement to this one. So the double crossovers are 1, big A, big B, little c, and 0, little a, little b, big C. Then we got to get the single crossovers. And this is why I say it's a good idea to get everything in the right order. So let me go ahead and cheat for you. And there's my little table of all these different results. So there's our non-crossovers, there's our double crossovers. Then here's the single crossovers. This is one category, which is big B, big A, big little b, big C, this one. And its complement, which would be this one, right? One thing to watch out for, notice these are kind of close. And you might say on the basis of just the numbers, that's one category, but it's not, because notice they don't complement, do they? But this one and this one do complement. So that's this one right here. And then the other one is Big A, big B, big C. This one, 25 and 15. These two complement. So that's our third category. So once you get them into these categories, then you can look at these and you can say, okay, this one represents a crossover between B and C, right? Because if we compare this to this one, for instance, there's a non-crossover. It's big A, little b, little c. This one is big A, little b. It starts off the same, but then C is different. So that tells us it must be a crossover event between B and C to produce this from this. Okay? And then that means this has got to be the crossover between A and B. There's our double crossovers. There's the numbers over there. And we can figure out the frequencies. So 92.9% are non-crossovers. This is pretty typical. You'll find lots and lots of non-crossovers. 30 out of 1,000, that means 3% are crossover events between B and C. 40 or 4% are crossover events between A and B. And there's our double crossovers, there's 0.1%. You gotta add that number to this number and this number to get the distances. Okay, so let's draw a map. What would the map look like? Like that. So notice the distance is 3% plus 0.1%, so 3.1 distance between B and C. 4.1 for B to A. Notice also, I intentionally drew it in reverse order because we don't care about the overall order. Either way works. Okay. So all clear? Yeah, you guys are going to be able to do these in your sleep soon. After spring break, I'll give you some more problems to do on your own. Okay. Oh, here's a messy one. Here's a multi-part question. And I am not going to give you any answers. You're going to have to tell me. So I'm just going to sit down here. But I'll just lead you through the whole set of problems. Okay, so I say, here we go. We got lobe. It's a mutation that's dominant. Big L, notice. And that changes the shape of the eye. And then there's black, which is recessive. Curved is recessive. And purple eyes is recessive. Okay, we got 4 different genes involved here. This particular problem is not a triple point cross problem. This is one where we go through pairwise for each pair of genes and figure out what the distance is. So, we take females from the F1 progeny, are backcrossed to their fathers. Oh, we got to step back here. Wait a minute. True breeding L females are bred to true breeding BL males. So low eye females, black males. What would the phenotype of the progeny be? Told you, I'm not going to tell you. Go ahead.
Big L, BL, BL. Big.
Wait, it asks for phenotypes, not genotypes.
Well, they'd have the eye shape of the females, and they would have normal or wild type.
Okay, right. So they would be low-eyed, and they would have normal body color. Very good, yes. So that's our phenotypes. Then what we do is we take females from this cross and we're going to back cross them to their fathers. So and that just means we're going to take these individuals and cross them back to these individuals. Okay. And then we get this result. Does that make sense? So in this case, the F1s are all expressing the dominant phenotype. It's just that the mutant here is dominant. So we got lobed, which is dominant, normal bodies, which is dominant. And we're going to cross those to an individual that has wild type eyes, which are recessive, and black body, which is recessive. Hey, this is a test cross, right? You just have to see that and then it becomes easy. So we're just doing a test cross with a heterozygous F1, and we get these results. So what's the map distance between lobed and black? I should ask first, what is the non-crossover category here? What phenotypes are non-crossover that look just like the parents?
L and BL.
Yeah, the most common ones again. So the wild type and low BL are rarer, so they are probably the result of a crossover event. So we can look at the probabilities there and ask, okay, what's the distance between L and BL now? How would you figure that out? Anyone? Let's see, they help you out just a little. So if we add those up, that's 600, 750 plus 250, that's 1,000 flies we're looking at. So we've got 1,000 total flies. So what's the map distance going to be? Anyone? You keep giving the answer. We're going to get somebody else to speak up. So we got a pairwise cross. So the distance is just going to be the frequency of the recombinants divided by the total number, right? So we got, this is a recombinant, this is a recombinant, that's 250 out of 1,000 flies. So what would the distance be? 25, yes, this is good. I'm glad somebody remembered their second grade arithmetic. Yeah, so it's just that. It's that easy. All we care about is just take the frequency of the rare recombinants divided by the total, and you've got a distance. Okay, we're going to do another cross. So this one is completely different. We're not looking at lobed and black anymore. Down here, we got PUCU female, so that's the recessive. So they must be homozygous if they're expressing that. And we're going to cross those to true breeding wild type flies. So what will the F1 flies look like? So PU and CU are recessive. The wild type is dominant. And in both of these, the males and the females are homozygous. So what will the progeny be?
Wild type.
Wild type. Yeah, so they're going to be heterozygous expressing the wild type phenotype. So that should be our answer there. Why, yes. And you can write it out in multiple ways here. Okay, so we got these individuals who are wild type, but but we know they're also heterozygous, right? Because we crossed this wild type male to mutant double recessive females. All right, so we carry it another step forward. We do another incestuous experiment. And we're going to cross these individuals. We're going to take the males of this group and we are going to back cross them to their mothers, who are right here. And we're going to get some F2 progeny. Okay, once again, what kind of cross is this? It's a back cross, right? With individuals that are homozygous recessive at both loci. So it's a back cross and a test cross all in one. So that's what we're doing here. And when we do this, we get these results. Okay. With the example of this one under your belt, you should be able to see this one is really easy, right? Recombinants divided by the total. And once again, we've done this clever experiment where we've gotten precisely 1,000 progeny, so we've got a total of 1,000 flies. And so now I ask you, what's the map distance between PU and CU according to this cross? Yeah?
20.
20, yes. So 90 plus 110 is 200 over 1,000, and it's got a map distance of 20. See, we're working through these step by step in pairs. So we got the distance between L and BL, and we got the distance between PU and CU. We have some other genes we want to figure out here. So anyway, your cruel lab instructor hands you a bottle of flies. Why is he cruel? Because he didn't tell you anything about these flies. He just tells you they're F2 flies. And they were produced by test cross. That's all he tells you. That's rude, isn't it? You should document all of your crosses, and he doesn't tell you. Okay, you count these flies. Again, this is the product of a test cross, and now we're looking at lobed and CU. Remember over here we looked at lobed and black, and over here we looked at PU and CU. Here we're looking at lobed and CU. Once again, we conveniently get exactly 1,000 flies. So what? First of all, the first question, what are the genotypes of the two parental flies? You know, that secret that the instructor didn't tell you. So what must the parental genotypes have been? How do you figure that out? Yeah.
The one with the most numbers.
Yeah, you just look at the two that are most numerous. There they are, 480 and 480. Those are the parental genotypes. So one parent was lobed CU and the other parent was wild type. Okay, so we got that for this part. Yeah, so there's our genotypes. Then the next question is, what is the map distance between lobed and CU? Again, this is just like the previous two examples. So we got a total group of flies of 1,000. Add those up, that's 1,000.
5.
Say what?
5. No.
No, not 5.
4.
4, yes. We got 40 out of 1,000, so the map distance is 4. Yes. Okay. So we've got 3 combinations, right? We got lobed to BL is 25, and we got PU to CU is 20, and we got lobed to CU is 4. We're almost there, right? So then finally, the cruel instructor, oh, he's not so cruel because he just tells you the next distance. Doesn't make you do any calculations, doesn't make you look at flies. He just tells you, hey, the map distance between BL and PU is 9. Now you have to draw a map. You got all the information there, right? How would you draw a map? You got a series of pairwise distances. This is like that classic Sturtevant measurement. Maybe it will help if I scribble it on the board over here. Okay, we got a series of measurements. The first one was lobe to BL. So there's L, there's BL. We're saying that's 25. The next measurement was PU to CU. And it was a little shorter, it was like this. There's PU, CU, and that's 20. And then we got lobe to CU is 4. So I'll put that over here. There's CU, lobe, and that's 4. And finally, BL and PU is 9. So let's put that there. Did I say BL and PU? Yes. So you got these 4 short segments. And I'm asking you to assemble those into a complete map. How would you do this? Yeah.
So L and BL, so L would be on the left, BL would be way to the right.
Okay, get that one.
And then CU would be next. And then
Oh, wait, but you don't have a connection between that segment and this segment. Or are you talking about this one?
Yeah.
Ah, okay.
CU.
So where would you put CU? I don't remember. It's got 2 possible ways it can go. It can go this way, or it could go this way, and you don't know which way it is yet. But that's the right direction. Yeah.
Would it go to the left because PU would be between L and BL?
So this PU is between L and BL. Oh, I see. So you're saying we're gonna put BL and PU here, but that doesn't get us all the way over. Oh, I see. But you're gonna say, okay. We're saying, okay, lobe, CU, and then
Could you switch
C.
that? Yeah, could you do that one where you switch it around from CU to PU?
and then you're gonna put that 20 over here.
Mm-hmm.
Does that work?
And then go from the PU to BL. That's
Okay, so then this would be 20 here and 9 there. Does that work? Maybe. Let's take a look. But you got the logic down. That's how you do it. You just piece it together. And I put BL. Does that work? Just that we're flipped around 180 degrees?
Or CU is in the wrong place.
Our CU is in the wrong
Maybe
place.
left. Left of BL.
Oh. Oh, so we're supposed to go, okay. So let's fix that. So we're saying there's PU, and then we got CU, so we should have, yeah, so then this is 20, right? I got No.
Sorry, outside of L.
Oh, outside of L. Well, this diagram is getting really messy. Let's just go with that one. But yes, you can see that's all you have to do is you sit there and you just sort of maneuver the pieces around until everything seems to fit. And then you're good. Okay. Should we go to the next one? Oh boy. So now we're going to do a triple point. Well, you're going to do a triple point cross. So once again, I sit down and take it easy. So I give you the data there. We've done a test cross. Here we're looking at V, W, and Z. And we get these phenotypes and those numbers. So first question, what's the arrangement of alleles in the F1s? And you're going to say, that's easy, right? Quick, jump in and answer the easy question. Yes?
The plus Z1 and the BW plus 1.
Yeah, and again, those complement. You check that, and that looks good to me. Okay, so that's our arrangement. Let's see. Yes, you got it right. Then the question is, which gene is in the middle? Yeah.
Z?
Z. You just randomly plucked that out? You just guessed? How?
Comparing the combination with just 1 and the combination with 367, the only one that...
Okay, so you compared this one with that one. And yeah, then Z is the one that's the same there, and the other two are different, so you'd say, it's got to be Z. So yes, Z is the one in the middle. Oh, these are getting too easy now. Now the fun part. But again, like I said, this is really helpful if you get this on a homework set or a test. Just take a moment and reorganize the data. So we got a couple things to reorganize. One is we got to list Z in the middle. So this has got to become VZW. And this one is, this stays the same, V plus plus. And this becomes plus ZW, you get the idea. Got to change all those around. And then also what it'd be good to do is, as long as you're rearranging everything, categorize them. We already identified the non-crossovers, those two right there. We can see the double crossovers are that one and that one. And then we have to group these other two. So, like I said, I'm going to sit back and wait for you to do that. I've got a table here, but I'll wait until you've had a chance to put it together, and we'll see if you get the same table I do. Some of you aren't writing, you're just doing it in your head. Oh, I wish I had your brains. So just let me know when you've got it done, and I'll show you my answer, which better be right. Everyone's got it done? Oh, some of you do. You're quick. Okay. I'll give you a little more time though. It helps so much to rearrange the data before you leap ahead and say, I know what this answer is. Are you ready for the big spoiler? Is it going to ruin the problem for you if I show it to you now? No? Okay. So there's what I got. So as you can see, I just rearranged everything so Z is always in the middle. This light blue here, that's the non-crossovers. This is one single crossover category. This is the other single crossover category. And this is the double crossovers. Is that what everybody got? Or something close to it? Okay. Good. So now you see this is helpful because what I can then do is I can say, okay, there's the non-crossovers, the single crossovers, and the double crossovers. And most importantly, what I can do now is figure out which single crossover it is. So this one I know is the crossover between V and Z, right? Because I can compare that to that. And how do I get this from that arrangement? I have a crossover between V and Z. And it will put that V right there. And once you get that, you can just say, okay, well, I could do the same thing for here, but no, I just know it's the other one. So that's going to be Z to W. Again, you look, these all complement each other within their categories, so that's all good. And there's the double crossovers. And furthermore, we know that this one is the middle one because you already explained it to us, that that's the only way you get this from this is if there's two crossovers to swap that middle one around. Okay, I put something else here. What else did I put here? I put some numbers. Oh yeah. So I added these up. So this is again out of 1,000. You will discover in the lab we never get exactly 1,000, but here it works just fine. So we got a total of 705 non-crossovers, 184 single crossovers between V and Z, 107 single crossovers between Z and W, and 4 double crossovers. So what are the distances? What's the distance from V to Z?
18.8.
Yeah. Isn't this easy? You just add that to that, and you get 18.8. And this one you add 10.7 to that same 0.4%, you get 11.1. There's our distances. So now you can easily draw a map, right? Yeah. Just something like that. Triple point crosses are really easy, right? They're kind of routine. You just go through these things. There are other permutations I can throw at you. I could say, for instance, I've used a sex-length trait. What other things have I done in the past to backfool everyone? I'll put together some more examples for the week after next, and you'll see. Okay, but in general, the outline of the method is straightforward. Oh, what is the coefficient of interference? How would you figure that out? Got one more question to throw at you. What's the formula for the coefficient of interference?
It's 1 minus the observed double crossover divided by the expected.
Yeah, so there's our observed. We know that one. That's 0.4. But what's the expected? How do we get the expected? You multiply those two single crossovers together, and that gives you the expected number. And when you do that, let's see, I'll just jump ahead there. So the expected is 0.188 times 0.11, or 0.02. We expected 0.02 double crossovers out of 1,000. So we should have gotten 20 out of 1,000, but we observed 4 out of 1,000, 0.004. So to get the coefficient of interference, you just take 1 minus the observed divided by the expected, which was 0.2, and you get a coefficient of interference of 0.8. And remember, coefficient of interference varies from 0 which is no interference at all, to 1, which is lots of interference. So this is saying there's a fair amount of interference going on in this cross. Okay. Oh man, that turned into a complicated slide. All right, let's do another one. Now this is the one I sent you. And this is getting you warmed up for the next experiment. So, right now you're setting up the cross of white miniature forked by wild type. And you are then going to get your heterozygotes, your F1s are going to be heterozygotes when you get back from spring break. And then you're going to breed those together. It's going to work because it's sex linked, so the males will all be hemizygous for the recessive traits. And and then 9 to 10 days after that, we will get our F2s. So here's some F2s. And I am showing you the data from last year. Yeah, we did this experiment last year too. And I'll just mention this. That group got 9,457 total flies counted. You're going to do better, right? You're going to break that 10,000 fly mark. Don't look at me like that. Yeah, we can do it. It'll work. Okay. So anyway, we got these. This is the total distribution. And as you can see, there's interesting things here. We're looking at females. So we segregated by that. So there isn't 8, there's 16 categories here, just like you will be counting in the lab. So one of the first things I ask you to do is just chunk the data together. Okay, this is kind of messy to work with. So sit down and figure out how to group it together into the non-crossover, single crossover, the 2 single crossovers, and the double crossover categories. How would you do that? For instance, just tell me, what are the non-crossover data in this set? You can see some that are most numerous, like that right there, right? So that's non-crossover. What else is non-crossover, though? What about this one? What about, let's go down here. What about that one? There we just distinguish between males and females, right? So this is a non-crossover, this is a non-crossover, this is a non-crossover, this is a non-crossover. So when I say chunk the data, I mean add up those 4 to make one non-crossover category. So I sent you this data. So has someone already done that? Or you lumped them together? We'll get to work. And then I will show you the, let's see, I know I did it. Yeah. So what I want you to tell me right here is just show me how many are the non-crossover, how many are the single crossover between miniature and forked, single crossover between miniature and white, and how many are double crossovers. Yeah, make those calculators sing, or your spreadsheets if you're doing it that way.
We can't edit the document, so
You can't edit the document?
no. We can only view it.
Okay, I screwed up. All right, well, can you tell me how you would do it if I had given you editorial abilities on that spreadsheet? What would, for instance, be the double crossover types? I'll point them out if you tell me.
The 93 and the 96.
So we got 96 and a 93.
And then 194 and 177.
And then that one and that one. So you would just add those up, and that would be your double crossovers. And then, for instance, the single crossover categories, like here, this one, This one must be a crossover between miniature and forked, right? So you have to identify all of those. And you chunk them together. So we end up with 4 categories out of 16. You still want to look at this, because as you see in a moment, we're going to take a look at the data and see if it tells us anything. Okay, so there, I chunked it for you. So those are the numbers last year's class got. And also further, if we looked at which one was which, this single crossover is between miniature and forked, and this one is between miniature and white. So we got a bunch of numbers here. And then I would ask you, okay, what's the distance between miniature and forked? And you can get all that just from these data here.
Is it 18.2?
18.2, no.
I did my math.
Is it 22.8?
Yeah, we've got 3 different answers here so far. Okay, let's go over the method, and we'll see where the slip in the calculator came from. So we want to get the distance between miniature and forked. That would be 1,724 plus 560. Again, you got to add the double crossovers.
Didn't do that.
That's the problem. Okay, yeah, you always got to add that, and then divide that total by 9,457. 0.24 or 20, yeah, or 24.2 map units. Yeah. Okay, so that works. 24.2. What about between miniature and white? It's the same procedure again. You take that number, add that number to it, divide by that big number.
26.7.
26.7, very good, yes. All right, so that's the map they came up with, and we'll see in a moment that wasn't exactly right on. So before we move on to this next related problem, think about the data. Look at the data there. You're gonna have to do this when your experiment is complete. Is there anything that jumps at you? What about the male and female numbers? What should the numbers of males and the number of females be?
Roughly the same.
They should be roughly the same, right? Do you think they're roughly the same?
With more females.
Yeah. If you add up all of these down to here, add those all up, that's the number of females. Add these all up, that's the number of males. Now when you do that, look at there, the ladies win. We got a lot more females than males. How would you explain that? You're writing the discussion of this lab report, which we'll eventually have to do. And you have to explain, okay, why do I have this disparity between males and females?
Something that's sex-linked.
Yeah, this is a sex-linked trait, but should it affect this? You just told me a moment ago we expect 50-50, right? Speculate, why didn't we get 50-50? Maybe it's because males are weak, right? They're all hemizygous. They've all got these mutant traits on their X chromosome. And maybe they just can't cope as well. So the females, yeah, we got more females because they can be heterozygous in some cases for some of these, so they're going to be doing better. So we're going to see a difference in the sexes. Do you think that could screw up our results? Could. We'll see. I also ask, okay, if we just look at the individual mutants, Remember, this is something I've told you over and over again about these. You can break them apart into the individual components. So what if we just ask, how about the numbers of wild type white versus mutant white? What should the ratio of those be? You know, the expected ratio.
White eyes is a X-linked trait, so maybe the males would have a higher number?
You'd think? No, it's actually because of the nature of the cross. Remember, this is a test cross. Remember we looked at simple Mendelian traits that always came out 1 to 1 to 1 to 1? Same here. We should get 50-50 white-eyed versus red-eyed in our group. And again, what we can do is work the data and ask, did that happen? So for instance, this one's got red eyes, this one's got white eyes, red eyes, white eyes, red eyes. We could go through and just add up all the ones that have a plus in the first position and all the ones that have a little w in the first position and ask, is it 50-50? And we can do the same thing for miniature and the same thing for forked. If I do that, yeah, so what I found was when I looked at these data that way, we've got 4,272 white-eyed flies in this collection out of 9,457 total. That's less than 50%, right? So white wasn't doing quite as well as red-eyed flies. And again, I'm going to have you guys go into Flybase and research these different alleles, and we'll see that again it's pleiotropic, that white affects a whole bunch of different phenomena in the fly. So maybe some of them reduce the viability. Then I did the same thing for miniature. So like this one's miniature, this one's miniature, this one's miniature, etc. I added all the ones that have M in the second position, and I got 3,922, which is even lower proportion than white. Again, it should have been 50-50. So miniature seems to be affecting the flies. They're not doing as well. And then likewise for forked, I did the same analysis there. And look at there, there should be 50-50, and it's more like 3 to 1, or 1 to 3, I should say. So we're not getting, or 1 to 2, yeah, that's more like 1 to 2 here. So the forked are not doing really well at all. Alternatively, what's another explanation? You've looked at forked, right? I told you they're hard to score, harder than the other ones. So this could also be an artifact of students not being as good at recognizing forked. That's a factor. But do you have any problem recognizing white eye versus red eye? None at all. So there are some real effects on the phenotype on survival in these flies. So we'll be looking for that. Okay. Next part. Oh, this will be different. So if you go to Flybase and you look up those genes, white, miniature, and forked, here's their locations. It's on chromosome 1 at 1.5. M is at chromosome 1, position 36. F is at chromosome 1, position 57. So what are the distances between the genes? This one is not tricky. This is just basic math. Yeah?
34.5 and 21.
Yeah, it's that simple. So we go into this experiment knowing the expected distance between these genes. And then we do the experiment, and I'm asking you something a little different. If we looked at 9,457 flies, what would be the expected number given these map distances? How do we solve that problem? I'll give you a big hint. We're going to do that triple point calculation in reverse. Okay, so when we do it in reverse, we know the distances between white miniature and miniature and forked. So what do we expect the double crossovers, the frequency to be? To have a double crossover, you're going to have a crossover between white miniature and a crossover between white and forked, or miniature and forked. So the likelihood of both of those things happening is going to be the product law. Remember that? So the probability of both of those happening is going to be 0.345 times 0.21. So we can already estimate what the number of double crossovers should be, assuming no interference, okay? So what we would do is we would expect the double crossovers to be that times the total number of flies that we're looking at, 9,457. So we should have expected to see 685 double crossover flies. This still applies to you guys. You know, we expect that proportion, 7% of the flies that you look in the F2s should be double crossover flies. What about single crossovers? What frequency do we expect to see of white and miniature crossover events? Just this one right there. How would you figure that out? Again, we're doing the triple cross calculation in reverse. And remember that we figure out the frequency that we observe of single crossovers when we add the double crossover frequency to it. You look like you know what you're doing. What?
So do you have to subtract the 7?
Yeah.
From the 34.
Exactly. So we expect 34.5 minus 7%. And that would give us the expected frequency of single crossover events. And we do that. And we would expect to see 2,577 single crossover events. And then the same thing with the miniature to forked. Just take that number and you subtract the double crossovers in this case. So we expected 1,301 double crossovers, and then the number of non-crossover events is just everything else, whatever's left over. So those are the numbers we expected from this cross. And here again, there's what last year's class observed, and this is what they expected. We did that calculation for them right here. Are the observed the same as the expected? You're saying no? How can you say no? You don't know yet. How would you test that?
Chi-square.
Yeah, we have to do a chi-square. I'm not going to do it here, but yeah, what you will have to do in your experiment is do a chi-square analysis comparing those. And I can tell you right now that they are way off. Way far off. And I haven't figured out all of it just yet, because some of them are kind of close. They worked out all right. It's just a couple of the numbers, I don't know what's going on there, but it's been consistent from year to year. So I'll be really interested to see what she gets there. So another thing you can do in the analysis of the mapping cross is you can ask, okay, my results are clearly different from the results that would be published on Flybase. Are they different from previous year's experiments? Because right now you've got all that data. Look at there. Okay, you see how to do this, right? You just do that triple point calculation in reverse and you get a set of expected numbers, and then that lets you do a chi-square analysis. which will tell you, I'm pretty sure it's going to tell you that your results are miles off, but that's okay. Because as I just explained in the previous slide, there's all kinds of other factors we can take into account to explain that. Okay, I'm going to post this on the Canvas site so that you can refer back to it. And then you are going to go have a fabulous spring break.
Are we gonna have a problem set?
No, I'm not giving you any homework or anything over spring break. But I will get you a problem set when you get back.